Okay, some people are still joining, but I'm going to go ahead and introduce you, Dr. Abdulkadir. Most of you, if not all of you, know Dr. Sarki Abdulkadir. He's the vice chair for research in the Department of Urology, and he's also the John Grayhack Professor of Urologic Research. He's a professor of urology and pathology at Northwestern. His lab is focused on understanding the molecular mechanisms that drive cancers. And he's been doing a lot of not only very strong translational research, particularly targeting MYC in cancer, and I think that's going to be coming to the clinics hopefully soon. And so we're really glad to have you at our conference, and looking forward to your talk.
Great. Thank you very much. Let me know if I'm not clear or if I fade out. But thanks for that introduction. So, you know, I thought that maybe for this internal audience, you know, I will kind of carry you through our journey targeting MYC, and especially emphasizing the sort of conceptual difficulties that kind of surround the issue of targeting MYC being an undruggable, what people call an undruggable target, and really like the sort of things that I think one needs to kind of pay attention to in trying to get an inhibitor. So for background, I have kind of a limited background about MYC in cancer in general, just so that I can have time to talk about kind of what we have done. But I think most of us are aware. MYC is a very well-known oncoprotein. There are three family members, three different proteins: N-MYC, L-MYC, and C-MYC, which generally we call MYC, that are expressed slightly in different tumors. But MYC is really the dominant one. So a lot of compelling data have established that these proteins are major drivers in human tumors. Okay, so they're dysregulated, maybe sometimes by amplification. Other ways of increasing messenger RNA without amplification of the DNA, stabilizing of the protein, and many different pathways. They are tumorigenic, and they are required for tumorigenicity. And even when the initiating oncogene is not MYC. So, for example, when you have a mutation in RAS, in pancreatic cancer or lung cancer, oncogenic RAS, you get a cancer. If you remove MYC, if you delete MYC, those cancers will collapse. So even though, even when it's not the initiating oncogene, because this is one of the hang-ups when people think about targeting, because classically what you think is, so you have the BCR-ABL translocation, you have a drug against that, you have a KRAS mutation, you have a drug against that. In the case of MYC, it's clear that even if it's not a driver, because it's important downstream, it can be targeted. And so there are many studies that show that these proteins are valid targets for cancer therapy. And very important, I think I have a slide to talk about the fact that, this inhibiting MYC could be tolerated because of concerns about normal tissue. So for prostate cancer, I have this kind of really bare bones kind of slide to lay out where these proteins play a role. As you go in the transition from benign to primary and, homosensitive, and then, castration resistant, and maybe now, if you put NAPC as therapy resistant, end stage neuroendocrine. So c-MYC is involved across the board, okay, across the spectrum. And then there is evidence that in CRPC you have further amplification of the gene, so further increase in the protein. And in the more neuroendocrine type, N-MYC is classically involved, and L-MYC has been implicated by, I think, very few studies. I don't know how solid is that now in primary prostate cancer. And there is also crosstalk with androgen receptor. For example, there is evidence that MYC regulates the splicing to promote the AR-V7 variant, and so on. And we have done some studies showing co-regulation of MYC and AR on common target genes. We and others have done that. But so in terms of thinking about MYC, it's a transcription factor. Okay, this is a crowded slide, but I think it's important for us to at least get the scope of the problem in terms of how we think of this protein. So we have MYC and MAX here in the middle. So MYC has a partner called MAX that is an obligate partner for it to heterodimerize and bind to DNA. Without MAX, MYC could not dimerize or bind to DNA directly. So you need MAX, okay? And then it regulates the number of target genes. This paradigm has been known for many, many years, okay? I just want to highlight that we know from a number of studies that many oncogenic pathways like hedgehog, notch, jack, whatever is your favorite pathway, most likely it can activate MYC transcription. So going back to the point I made earlier that you don't need to have amplification of MYC itself in cancer for it to be important. Other pathways also like the MAPK, mTOR, are involved in actually affecting translation from MYC RNA. So you end up producing more MYC by activating these pathways. And then again, others like the PI3 AKT pathway will stabilize MYC protein. So end result, all getting you increase in MYC. Now, the problem with the field for many, many years has been that this issue of what are MYC target genes was really kind of a big mess because essentially, people were trying early on to define what are the key or magic MYC target genes that mediate its function in promoting all of these processes, right? Tumor, which lead to tumorigenicity. And the fact is that it does many, many things. And sometimes in different cells, it does, it activates different genes. But clearly, and then these are direct target genes. So directly MYC will bind and regulate processes involved in cell growth, cell cycle. These are just some examples of the genes, cell survival, metabolism, DNA replication, and also increase RNA levels, so that the end result is that sometimes when you overexpress MYC, you have a secondary amplification of RNA. And this has been interpreted to actually say that MYC regulates all the genes that are expressed in a cell, for those of you who may be aware. But in any case, so this is kind of the biology. Any hallmark of cancer you look at, most likely you'll find MYC is important. And a lot of these processes are important for normal cells also. So there has been this idea for many, many years that, look, even if you get a MYC inhibitor, you're gonna, it's just gonna be so toxic to normal tissues that you're not gonna have any therapeutic index. So a lot of what, I personally and our group have been kind of interested in as we do this work is really thinking about this idea and, can there be a therapeutic index? And if there is, what is the mechanistic basis? Why a protein that regulates so many things, how or why could it be selected, if it is selected? Okay, so targeting MYC directly has been a challenge. And so this is just a cartoon showing you, like, if you have the MYC DNA gene, it's transcribed, you get MYC RNA, it's translated, you get the protein. And then the protein could be stabilized or activated by, let's say, phosphorylation and other things. And then the MYC could complex with MAX and function. So inhibiting MYC directly by, a small molecule has been really a challenge. So over the years, people have done different things. Maybe the most well-known is like the JQ1 bromodomain, right? Which basically in some cancers, the enhancer of the MYC gene is dependent on bromodomain protein. So if you inhibit that, then you shut down the RNA and then you don't get protein. But again, like I said, this is only in some cancers and in some cells. And then there are many other different kind of indirect approaches. So all of these are good, but they are indirect. And so whatever the end result of what you get depends on what else is going on with each of these targets that you target. So there has been a long-standing interest to directly target MYC so that you can isolate exactly what's going on there. And not just for therapeutics, but also to understand function. So I think I have probably I accidentally missed some slides, maybe because when I transferred this to for this presentation, but I can give the talk without those slides. So what we did is to revisit. Okay, so over the years, what people have done early on was to set up assays that you find molecules that disrupt MYC-MAX interaction. Okay, reason being that MYC is disordered, and so there is really no nice pocket where you can design a small molecule to bind. So you cannot do that kind of, structure-driven development of inhibitors. Okay, so what people have done many years ago is to say, you set up a yeast-2 hybrid system where you set up an assay where if you screen compounds that disrupt MYC-MAX, you get a signal from the yeast-2 hybrid system. And using those kind of approaches, people have found a number of small molecules that are able to disrupt MYC-MAX interaction. The problem was that basically most of those, or almost all of those compounds, were not really chemically very suitable to become potential drugs. So they are rapidly metabolized, so you can't even get, can't even do mouse studies. And in fact, to be honest with you, one of the main motivations for us to do this work of developing MYC inhibitors was because from my prostate cancer studies, a lot of pathways lead to MYC, and I was really interested in MYC. There are no inhibitors we can use in vivo. I thought that we could do something to generate new compounds that we could use in animal models to answer, to really validate what MYC is doing. So what we did is to set up a screen in silico. And the basic idea behind the screen is that we looked at the literature to find compounds, chemicals that have very good evidence of binding to MYC. Okay, and we generated a pharmacophore of those, which is basically kind of integrating the chemical features of all those. There are about 30 of them. So we defined 5 different features chemically, the pharmacophore, that then we use to interrogate a chemical library in silico. Okay, and because this is in silico, we could actually the idea is to find compounds that have similar features, and because it's in silico, we can look at a large number of compounds. And this is the second point that kind of guided the approach. The hypothesis we made right in the beginning is that since there are examples of compounds that are able to bind to MYC and disrupt MYC-MAX complex formation, if you search, a much more vast chemical universe, you should be able to find similar compounds. And among those, some of them should be able to have features that allow them to survive the metabolic destruction in animals, in mice, so that they are stable in vivo. Okay, so we did that. We screened, initially we started with a library of 32 compounds. And then we removed about 60 million of those compounds because they just didn't have the chemical features of drug-like compounds. So now we know much more than like 15, 20 years ago of what drugs tend to look like. So we remove those. And then we found about 70 hits. Okay, they are all structurally different. And we screened them in vitro. I'm not going to go into that. But when we picked one of them, which we call Min9, and we then made a lot of analogs of that Min9, just make different changes, and we screened those, and we came up with the one that I'm going to first tell you about. Okay, so we have this compound called 361, and because of the chemistry we have done, we also have a negative control compound called 360, which is very, very similar to 361, but it doesn't bind to MYC, but they have pretty much the same atoms. They just a methyl group is just added to a different portion of a ring. But so all of what I have told you so far, the reason why I'm not even going into it is things that people normally will do. They will screen and find something, and then they will publish. But we wanted to really make sure that what we have are really good chemical probes that are really binding to MYC. Okay, and one of the big questions out there is target engagement, meaning if you have a small molecule that you find that you think binds to a protein, in this case MYC, then what is the evidence that this compound binds to your protein inside cells? Okay, not in vitro where you can have biophysical assays with pure protein and pure compound. Also, if you can do this without modifying the compound, okay, like adding a fluorescent tag, for example, that would be nice, because the fluorescent tag may change the features of the small molecule. So if you can do it without modifying the small molecule, that's better. And if you can do it with also modifying your target protein, in this case MYC, so the native MYC that's present in cells and the native, if you will, compound to show inside cells they interact. So Hui-Ying Han in my lab spent about 6 months working on this issue, on this assay, and it was finally able to get this assay working for MYC inhibitor. So this is a cellular thermal shift assay. It's a very simple idea that if you take cells and you heat them in a very controlled way, and then you run Western blots and you measure your particular protein, okay, by Western blot or any other method, the protein will have a specific melting curve. Okay, this is what it is. It's going to be denatured, right, with time, and this is very specific for the protein. If you add a small molecule that interacts with your target, it's going to change the biophysical properties of the protein. So now your melting curve can be shifted, either decrease or increase. Okay, and so we see that this compound 361, which binds to MYC, is engaging MYC inside the cell. The negative control compound 360 doesn't do that. The other thing we did is, okay, what are all the proteins that your inhibitor bind to? So we made the biotinylated analog of this compound, and then we did a pull down with streptavidin, okay, in cells. So you pull down MYC, so this is input. Importantly, you don't pull down MAX, because as I said to you, our whole screen is for compounds that disrupt MYC max. So once compound is bound to MYC, it's dislodging MAX. So you don't see MAX. So that's a very important point. And then we checked other proteins, kind of unrelated, like HIF1 alpha here is shown. It doesn't show any binding. But we also did mass spec proteomics, and this is published in the paper that Hui-Ying published, where we showed all what are the proteins that are bound. And most of what we've pulled down are proteins that are known to interact with MYC. So we think it's because MYC is bound to those proteins that you pull down. But we put that out there and we publish it just to disclose fully what this compound is. And I might say this is the first time anybody with any potential MYC inhibitor has really done this kind of studies to really look in an unbiased way what binds to. Okay. The other point that to just buttress the binding of the compound to MYC is by NMR. In this case here, we are looking, this is saturation transfer difference NMR. And bottom line here is you can see this is signals of the compound binding to MYC, but not to MAX. Okay. So other things we have done is to see inside the cells. Okay, when we say we can disrupt MYC max, and we have shown that in vitro. I'm not showing any of that, but in cells, when you treat with a compound, can you show evidence that you disrupt MYC max complex? So we do co-immunoprecipitation. In this example here, we pull down with a max MYC antibody. Okay. And you can see in the DMSO condition, you recover this much max. This is just controls here. So you recover max. Within presence of compound, okay, you recover less max. Okay. When you normalize to the total amount of the protein in both conditions, you can see you have a dramatic reduction in the MYC max complex formation. The other assay that one would like to do is a proximity ligation assay. This assay basically in cells or tissues, you have an antibody against two of your proteins. So in this case, MYC and MAX, and then there is oligo that's linked to the antibodies that can be amplified by a rolling cycle, and then you detect these red signals. So these red dots indicate that there is a lot of MYC and MAX kind of very close to each other, but when you treat with this inhibitor, you see you decrease that. So less MYC marks near each other in a complex. The other thing which we noticed, which this was not something we are expecting, is that these compounds will decrease MYC protein stability. And this is just showing you cyclohexamide's pulchase assay. What cyclohexamide does is stop new protein synthesis, basically. So then, you know, you are looking at the existing protein, and then over time you can see how long it takes for the normal cellular degradation pathways to degrade it. So MYC is a short half-life protein. In fact, in cancer cells, the half-life is increased. So you can see in this prostate cancer cell, half-life is like 52 minutes. In more normal cells, it's much shorter. But the treatment with the inhibitor actually reduces the half-life by, you halve it. So this is CMYC. We also see evidence that the compounds will lead to degradation of MYC. There is no change in RNA. That's why we call it degradation. I'm not showing those data here. So one of the things we did, which I think could be important in terms of thinking about a biomarker for this compound, is we decided to look in more detail into how this inhibitor, the MYC inhibitor, is increasing MYC degradation. Okay, so the pathway in which MYC protein levels are controlled in the cell are kind of well studied. This is one of the major pathways, and it's really kind of a neat system in the cell. So MYC, as an oncoprotein, you don't want a lot of MYC running around in the cell. Okay, you want, if you need to proliferate as a cell, let's say we get a growth factor signaling, and let's say ARC is activated, okay, then what happens is normally MYC is not very stable, and it's not even transcriptionally active until it is phosphorylated on the serine number 62. Then it becomes stable and transcriptionally active. Okay, and this happens all on chromatin. Okay, and the complex is nucleated on axin. So when serine 62 gets phosphorylated, now this MYC protein is active and it can transcriptionally activate target genes. But as soon as it has a signal, the GSK3 beta kinase, okay, which is also present on the nucleus, and sometimes it's activated by a different arm of the same growth factor signaling. So you get almost like a clock. The first hit you activate MYC, so it functions. Then later on you activate this, which will GSK3 beta, which will inactivate MYC. So that way you control the window of time in which MYC is actively transcribing genes. Okay, so GSK3 beta will recognize serine 62 and then will phosphorylate MYC on threonine 58. And then this threonine 58 now becomes a signal for degradation. So you can remove this S62 with a phosphatase, but the threonine 58 is what is recognized by the ubiquitin dependent proteasome degradation pathway, and then it gets degraded. So, you get MYC to function within a short amount of time. So what we did is to look at this pathway, right? So when we treated with our compound early on, before you start to get any effect on total MYC degradation, which started at 3 hours, we see the phosphorylation on threonine 58 is increased. The serine 62 is not changed, and GSK3 beta activity itself is not changed. Okay. And we can reconstruct this in vitro. Maybe I will have a slide, but the model we have is that the compound makes the MYC protein much more amenable to being phosphorylated by GSK3 beta, and then it gets degraded. Okay, you can see this quantitated. If you take the ratio of the phosphorylated threonine 58, the total MYC, you can see increase, and then the serine 62 doesn't change. Okay, and if you mutate the threonine 58 on MYC, okay, to an alanine, so it cannot be phosphorylated, now you cannot degrade this the protein effectively. So this is a major pathway by which our compound works. So we reconstituted this in vitro. We got a recombinant MYC protein, okay, and the recombinant ARC. ARC phosphorylates on serine 62, and then we add recombinant GSK3 beta in the presence or absence of our inhibitor, and then we monitored the 58 phosphorylation. As you can see, the negative control compound doesn't really lead to any increase in the 358 phosphorylation, but the 361 compound dramatically increases it. So we think this is a direct effect. One of the really nice things is that we can detect this in a robust way in vivo, because they are very high quality antibodies for these phosphorylations that can be detected. Here, if you grow these cancer cells in a mouse and treat with the compound and then take out the tumors, you can see a nice increase in the tyrosine 58 phosphorylation. So this could be a pharmacodynamic marker. When you give the compound, and you take out the tumor, you can see evidence that your compound has gotten into the tumor and has done its job, which is to interact with MYC and change its function by monitoring for the T58 phosphorylation as an example. Okay, so these compounds do all the things that you would expect a MYC inhibitor compound to do. If you look at RNA-seq, this is, I think, from a prostate cancer cell line. If you treat, if you look at the genes, the most dramatic changes occur in MYC target genes. Okay, so this is by gene set analysis. So, and individually, you can see all the MYC target genes in the cell cycle here as an example. They are all dysregulated in the right direction by the MYC inhibitor. So it definitely suppresses MYC transcriptional activity. How selective are these? Early on, we tend to use cell line that we use many different models, including MYC knockout. But one of the ones I really like is this. This is a pheochromocytoma cell line. Okay, so MYC is kind of really is required for the proliferation of essentially almost all cells, just to different extents, including normal cells. But pheochromocytomas, some of them have mutation in MAX, and so they don't require MYC MAX. Okay, and they proliferate normally. So we like using that as a negative control to show our compounds do not have an effect on that. They do have an effect on a lot of other cell lines like these prostate cancer cells and many others. We also have an inducible model where we can control the levels of MYC in a lymphoma cell line by using tetracycline. You can see as you increase tetracycline levels, the MYC is decreased. This is an engineered model. And what we see is that with this model, the sensitivity of these cells to our MYC inhibitor correlate, okay, are inversely correlated to the amount of MYC. So the more MYC you have, the more sensitive. The less MYC, the less sensitive. Okay. So and also we can generate organoids from normal mouse prostate here of the same background strain as the MYC cap. Which is a prostate cancer transgenic mouse model. You can see the tumor, the cancer organoids are much more sensitive. As I said, also we have MYC knockout cells, fibroblasts, and wild type, and we have used those as well. So in vivo, this compound that I've been telling you about, which was like one of our first leads, 361, is quite good at inhibiting tumors. So these are established MYC tumors in the FBB background. So this is immunocompetent, and I will come back to why we thought even from the beginning we liked using that for our screens. So you can see, this is how these tumors grow. Normally they grow, robustly. You do some high dose here, 70 milligrams per kilograms twice daily. I think this one is. Or if I'm not mistaken, you get nice response. You wait, the tumors will creep back, and then you can treat with a lower dose. And so this is 100 a day, and then this is 70. And when you look at the tissues, you shut down proliferation. You have some increase in CAS space. Okay. However, I want to note that we knew that this compound is quite potent, but maybe not very well tolerated, because we can see the condition of the mice and the body weight. with this higher dose, even with this dose, it's not, it's not going to be, it's not going to have a good therapeutic window. So we knew we had more work to do. Okay, but let's come back to the issue of because of the literature where MYC plays a role not only in the tumor cells but also in immune cells. So there are papers out there which says maybe MYC is important for NK cells or T cells or B cells. So I was thinking to myself, if you get a very good inhibitor, MYC inhibitor, what if while you are inhibiting tumor growth, you are also inducing immunosuppression because you are wiping out T cells or B cells or NK cells or whatever, because MYC is just kind of everywhere. So because of that, I wanted to make sure whatever we get right off the bat, we test it in the immunocompetent mice so you can see the net effect, which is what I just showed you, that in that model, which has, it's not an immunodeficient model, it's immunocompetent, whatever our inhibitor is doing, the overall effect is good because we are getting inhibition of tumor genesis. Okay, so that's one of the reasons why we did. The other reason is this, okay, which is kind of summarized here. This is summarizing a lot of different studies, but I think this makes the point nicely. So, people can make models where you can turn MYC on and off in a transgenic mouse, in the liver or in the lymphocytes or, different tissues. And what you see is the same thing all the time, that if you do that, you activate MYC, you get a cancer, it's an oncogene. You get a tumor. If you shut off MYC, then you get nice tumor regression. And you see that what happens is, you have apoptosis, you can see decreased angiogenesis, you can see senescence, all of these things. But it was clear to people that you need an intact immune system to do that. Because if you do this in the background of immunodeficient mice, right, like rag knockout mice that don't have, like the T cells and B cells, whatever, you can get the tumor, and when you remove MYC, you get a response, but it's not as robust and it's not as sustained, indicating that you need the help of the immune system, okay, to clear out the tumors. And this shouldn't be a surprise. And I think this probably, for many anti-tumor agents, just like when you have an infection and you take an antibiotic, having an intact immune system kind of helps a lot to clear the infection. So it's the same kind of idea. So because of all these reasons, we then looked in the tumors from where we treat with our inhibitor. Initially, we just look at CD3 T cell infiltration. We can see a lot of T cells come in, and to our surprise, we also see a very nice, robust upregulation of PD-L1. So these tumors, normally don't have any PD-L1, and we have shown in some other studies that they don't respond to anti-PD-1. But now with the treatment of the compounds, now you get this upregulation. And by flow, we looked, and this is some of what we looked at. You can see you get a lot of CD3, CD4, CD8 NK cells, gamma interferon positive CD4 cells, and IL-4 producing CD4 cells, and many others that I think this has been published. Most of these changes are consistent with, nice inflammation happening. You have recruitment of these immune cells that should be helping to clear the tumor, okay? And we looked in the more distal lymph node, to see if there is like a general effect on the immune system. And we didn't see. We have done a huge panel of the same markers, and we didn't see so far anything. We will need to look more closely to see if there are more subtle changes, but we didn't see any evidence. So it was very nice, very encouraging, because at least we can get an anti-tumor effect without kind of shutting down the immune system in general. So what is the mechanism? Why do we get this infiltration and upregulation of PD-L1? This is the model we think is happening. We think that our compound is killing cells by the mechanism of immunogenic cell death. basically, and this is the evidence, part of the evidence for that, because in vitro, if we treat these cancer cells with our compound, we get a release of HMGB1, release of ATP, and cell surface calreticulin. And what are these? These are your classical danger dumps signals that when they are produced, they do have receptors of immature dendritic cells. So basically, what they do is they activate the immune response. Okay, so then you get now production of cytokines and recruitment of a lot of the cells that I showed you earlier. And because of the cytokines that are produced in the area, you can now induce PD-L1 on the tumor cells. So we think that's why we have the induction of PD-L1, because in vitro, our compound doesn't directly cause upregulation of PD-L1 in the tumor cells. Okay, so this happens in vivo only. But so anyway, so what this suggests is that, okay, if you have anti-PD-1 resistant tumor that doesn't even express any PD-L1, you treat with the inhibitor, because of what I just told you, you get upregulation of PD-L1 on the tumor cells. Now maybe you get a response to anti-PD-1. Okay, so we set up to show that, and that's what we see. So this is like using suboptimal treatment with the inhibitor compound, like 2 days on and then 2 days holiday, 2 days on, and then PD-1 also twice a day in alternating. And you can see the single treatments didn't have an effect, but you get a nice nice tumor regression actually while they are on the treatment. And this some of the mice didn't, the tumors just didn't grow back. So I think with the optimization of the dosing and whatever, we can probably get even a better response. But this made the point that there is synergy between the MYC inhibition and anti-PD-1. So coming back to the fact that this 361 compound, I told you, is not very well tolerated. So if you look here, we did acute toxicity. We can tolerate, mice can tolerate up to this much, to 40 milligrams per kg. But we made a new analog, which is much more tolerated. We went up to 1,000. Mice are fine. Actually, we couldn't go more just because we couldn't give more due to solubility. And this is a compound we call 975. In vitro, and it has the same activity. Actually, it looks better. It doesn't affect the PC12s. It affects your MYC dependent cells. We can do chronic treatment of mice with that, and we have normal CBCs, LFTs, renal function, everything. And then we had a veterinary pathologist actually, look at all the tissues that we took, and they really didn't find any abnormalities in the trees. So this compound can be well tolerated, and this profile is actually reflected at a molecular level, because if you look at RNA-seq, so look at this top part first. This is RNA-seq of PC3 cells treated with either the 361 compound or the 975 at the IC50 for each compound for 24 hours. Okay, you can see the first thing is that the 361 compound basically affects many more genes than 975. There is a very good overlap between the two, and the 361, now we begin to see some genes that are not seen in the 975 that are affected, like this, a lot of histone 1H genes that we don't really understand why, and some electron transport chain genes, and maybe this is why this has less more toxicity, if you will. But then for the 975 compound, we want to look using this assay to see how selective it is. Okay, so we then did this experiment. So this is RNA-seq from an inducible cell line where you can induce MYC levels. So this was published by Chidang, and they did a duplicate experiment and did RNA-seq, and they found this number of genes, the one in the pink here, that are all regulated by MYC. Okay, so we got the exact same line, and we did the exact same conditions like they did. That's the tetra And the only thing is we did more replicates. Okay, so we got less number of genes, but then there's a huge overlap. Okay, and then if you treat the same cell lines with 975, you get the same overlap as when you do the same cells in different cities, you know, in different labs. So bottom line is our compound by RNA-seq mainly affects MYC target. There is a small this cohort of genes that are unique to the small molecule, and when we look at those, they are all the pathways that are enriched there have to do with response to organic compound. So because these other studies, the tetracycline is in aqueous, it's in water basically. I bet if we were to put tetracycline in the same you know, in DMSO and did this experiment, a lot of this will also show up in the other one. So bottom line is we think this is very specific compound. Okay, PK levels are good. You have a good half-life here by IP and by oral. This compound and we've done IP, oral, and IV, and the amount of compound you get is enough to, you know, it's multiples of the in vitro IC50, so it's consistent with the activity that we see in vivo. We have more recently now done drug levels in the tumor, and actually the drug levels in the tumor is like twofold. Although this one, the experiment was only 3 days treatment, but at least in 3 days treatment, we have twice the amount of compound that's in the tumor than in the plasma. Okay, so the PK levels are quite encouraging. This 974 compound has efficacy just like the 361, and basically it does all the same mechanistic things I showed you for the 361, because the reviewers actually had us do all of those things again for 975. So, you know, induction of 250 addition, example here, PDL1, the synergy with the anti-PD1, and many more things also now that we have done with this new compound. So at this point, here's our summary that we have this new compound chemical probes that will bind to MYC and disrupt MYC-MAX binding, so that will affect the ability of MYC to transcriptionally activate its target. At the same time, they make MYC more susceptible to phosphorylation by GSK3 beta, which is a signal for degradation. And that this agent, you know, can inhibit tumorigenesis in vivo and will lead to recruitment of a lot of immune cells and upregulation of PDL1, which now makes these tumors set up for responding to anti-PD1, where you see synergy. So the next kind of maybe few minutes, 5 minutes, I want to just highlight one more point about this compound. So this relates to, you know, why, how can we, you know, how are these inhibitors functioning to allow us to see this kind of nice therapeutic index? You know, why are we not shutting down all the target genes in normal tissues and so on? So this goes to this idea that, you know, if you think about the target genes that are regulated by MYC, these have 4 examples here. You increase MYC levels, what you get is that there are some of these genes which have high affinity binding sites for MYC. Okay, so to this example here. So under normal physiological conditions, you kind of get, these are the genes that you are regulating. So these are your core target genes, okay, that MYC is designed to regulate. As you increase MYC levels, like in cancer, now, you know, extra MYC could recognize maybe low affinity binding sites and regulate them either to activate or to repress, depending on how the function goes. So this is an idea that kind of has been floating around, that, you know, this, you know, the oncogenic MYC and physiological MYC may be different. So accordingly, you know, I was thinking that, okay, if you have an inhibitor, depending on how your inhibitor is functioning, you may actually be hitting these new targets that are recognized, okay, preferentially. And so you don't need to actually wipe out everything come here, because if you come here, then, you know, the mouse is dead. So maybe you really want to be here, and is this what we are seeing with our compound? Okay, and so people have looked at that. But so we have done some RNA-seq and ChIP-seq experiments by Austin Hobbs, a graduate student, and collaboration with Debu. What we see essentially is that indeed this is happening. So this is ChIP-seq, okay? This is MYC. Across the genome, you can categorize at least two classes of target genes, one which we call like high affinity and the low affinity. And you can see the low affinity genes are preferentially where you lose MYC binding with your MYC inhibitor compared to the high affinity. You can see this more clearly here. You can see this is the MYC binding here in the promoter of this MCM10 gene, which is involved in DNA replication. And you can see you are wiping out very nicely the MYC binding. And this is the RNA-seq tracer. You can see you suppress that. And these are all the exons here. You lose the expression. By contrast, this target, which is, all these are non-canonical validated MYC targets, by the way. You see a lot of MYC binding. You still have quite a lot of MYC binding even with drug, and you can see you are not touching the expression. You have nice, robust expression of this target. So this is a high affinity target. Okay. So what is really nice about this is when we looked at all these type of genes and we did gene ontology analysis. This kind of low affinity targets, a lot of DNA replication, DNA repair, cell cycle, all the things that you want to inhibit in cancers, right? These ones are mainly ribosome biogenesis. Okay, so a lot of like maybe 50 ribosomal genes, which are all MYC targets, but they are not affected. So we feel that maybe this could actually provide a mechanistic rationale for kind of what we are seeing. We have done a lot of, and some of this is, done in collaboration with some of the, pharma, preparing rather for with Invoke kind of helping us navigate this. So a lot of medicinal chemistry, phosphorylated compounds, which we have identified with that are more potent, and Polsky actually also has really helped us to do that. We are now about to do PK on 10 compounds that are all more potent than this 975 compound that I showed you, and we've been validating them. We have been doing some combination immunotherapy, anti-androgens, and PARP inhibitors because of this issue that I told you, that this DNA replication and repair issue here. And we have some mechanistic studies that we have a new R1 to look at that I haven't talked about here on really understanding some another novel aspect of these compounds in terms of what they're doing, and this kind of more development aspects of this that I think you might want to know where we are with that. Really, we are now on the cusp of selecting what is going to be our IND agent, so we can do the IND enabling studies. We have done some work on formulation. We have a good oral formulation for animal studies. We have done more of the PK/PD, including tumor drug levels, like I just said, told you. The synthesis has been scaled. Fortunately, it's only 5 steps in the chemistry, and from what the chemist told me, that's good. So it's not going to be a problem making this, and we have this project also as part of our spore application, and definitely we have an interest in maybe doing some early clinical studies, because the big point is that there have not been direct MYC inhibitors that have gone to the clinic. So I think we can learn a lot in terms of, when you're giving the compound, are you getting target engagement? Is it behaving in the human like the way it behaves in the animal? I think, that's extremely important for us to learn about that. So many thanks to many collaborators from our side. Hui Han really did a great job of kind of sticking with this project. She's just been recruited by AstraZeneca. Mihai, Barbara, and a lot of people here that I have listed. Gary Shields has been, the medicinal chemist who has worked with us on this, with Atul and Habi. And the early computational chemistry was done by Rama Mishra, and now we are collaborating with Debu and his group on some of the genomics. A lot of support from Northwestern, from the Polsky to the Cancer Center, and the New Cures Accelerator, and the PCF, and the SPO. And now the R1 will allow us to do some additional, more basic studies. So this is kind of our story, and this is where we are. So I will be happy to the question. Sorry, I think I went a little bit longer than I hoped, because I always like to leave more time for questions. But any questions, feel free.
Sarki, that was it's Ted Schaefer. That was awesome, and thank you for presenting that to our group. I have a quick question for you, which is broad. You glossed over a little bit about some of the innovative approaches you took to actually identify these compounds, but I wanted to know what your thoughts were about this basic approach and how you could potentially use it to identify other compounds that are either of undruggable oncoproteins or other kinds of targets.
Yeah, I tell you, I was shocked when several of my slides were missing. I don't know how it happened, right? Somehow in preparing this from yesterday to today, some slides got deleted, or maybe when I transferred from one thing to another. So you are right. So here's the approach, right? What is innovative? So the in silico screen, people do that. One aspect, I think, is that screening a large number of compounds will allow you to find all the different interesting compounds that could actually do the job that you want to do. But that's not the most innovative. I think the most innovative part is when we did all the in vitro screen, right, in silico and in vitro, and we tested and we made even new analogs that are more potent. This is very early on, and we are really excited, right? And we did the microsomal stability that people do in vitro to show that your compound is not destroyed by the liver, microsomal enzymes, all that stuff. And then we went to in vivo, and it was negative. Okay, the PK showed that it's not active. So I think, especially for academic labs, to really go through that, each compound, do the full PK and that's just not feasible. So what we did is we set up this rapid in vivo screen, okay? And what it is we made a cell line, actually it's the MICAP cell line, which has an E-box, okay? That's the MIC-responsive DNA element driving luciferase. So once you inhibit MIC activity, you will see decreased luciferase, even if before you see a decrease in tumor size. So you should see that within it. If you have an inhibitor of MIC activity and you have a mouse that has these MIC cells growing in them, you treat within 2 days at most, you will see a decrease in luciferase signal, right? And we can do fewer mice and we can batch them. And so, when we did that, we found that 85% of all the compounds that look great in vitro are not good in vivo, maybe because they are rapidly metabolized or whatever. We didn't try to figure out. So only 15%. Okay. So this is like how to get over the hump. So I think what I will say is that when thinking about, any, when somebody's trying to make a new inhibitors, whatever, kind of thinking, of the mechanism and how to exploit it to get to the end game, which is at least you want something that works in animals, right? Because if you have a compound that doesn't work in animals to inhibit anything, why use that? You can use siRNA or CRISPR to remove your gene. So you really want something that works in vivo, and I think that is the thing which we kind of tried to highlight as something new. So we integrated this into our assay, and then we came back alive, because the project almost ended at that point, because we couldn't have gone forward spending a lot of money and time and finding negatives after negative.
Sarki, I just had a couple questions. First, I'm not sure you said it, but so do you need an intact immune system for your compound to work in vivo? I thought you implied that, but maybe I misconstrued it.
Well, no. To work maximally. So we have actually done, a bunch of experiments. These are all in the paper where we actually take the same cell line, grow the same tumors in either the syngeneic background or the immunodeficient, and you can see, you get a boost obviously with the immunocompetent, but you can, this inhibitors still inhibit proliferation and other things, so you can suppress tumorigenicity, but clearly, especially if you are trying to get regression, it needs to be in the immunocompetent background. Yeah. How much of it is the, I guess one question we've always asked ourselves is, and we talked about it a lot in our group, like how much of the activity is contributed by the immune system, if you will, right? We haven't nailed that down, and I really can't tell you, but for full activity, you need the immune system.
Yeah, actually,
Sarki.
I don't know if it's true, but the feeling, some of the immunologists in melanoma feel that the targeted agent, BRAF, MEK target, do not work in an immune deficient mouse. but I've never seen that data myself.
Yeah, that's interesting. Yeah, no, I mean, yeah, go ahead.
I was going to ask you, just as a follow-up to Jeff's question, have you looked at the high affinity versus low affinity targets in the immune compartment specifically, as opposed to that?
Right.
Could be pretty cool, actually.
Yeah. So that actually, that's exactly the thing we're going to do. We are going to be now directly testing these inhibitors, and we'll take one immune compartment and study this issue. Yeah. So we will do that. That's a good suggestion. Yeah.
What do you, Sarki? Maybe the last question. What do you anticipate might be dose-limiting toxicities of the of the drug? As we go forward towards clinical studies, and also I couldn't tell from some of the slide prints. What type of dosing are we thinking about?
Okay. Yeah. So first of all, regarding the toxicity, I think that still we need to watch out for the gut, right? Because the rapid turnover of the cells, so a lot of you need a lot of proliferation. A lot of that is driven by MYC and the hematopoietic system. Again, the level we are seeing, the level we have used so far for our mouse studies spares those tissues, because like I said, we did the full autopsy and we didn't see anything there. But I think that that's what's going to be dose limiting. So in a way, it's nice because MYC is so well studied that you kind of know all the normal systems where it is involved. So really one could look at those, right? So these are the big ones. In terms of the dosing, where we are right now, especially when we have been testing a lot of different tumor models, and these are all immunodeficient, so we are trying to see like which tumors are going to be. So what we found is that we twice a day dosing, oral dosing. So right now in the mice we're using 100 milligrams per kg oral, that we get very nice tumor growth inhibition with that. So this is even in the absence of the immune system, because these are all immunodeficient mice. So this oral is good. We looked at tumor levels with the oral dosing. This is the one I mentioned, that you get like twice the blood levels in the tumor with the oral dosing. So definitely the route, I think, could be oral. As you move along to the translating for human, I think, with input from all the PK people, maybe once a day or twice a day dosing could be accept fine.
Wonderful. Thank you again for a great talk. For everyone on the conference, we didn't put up the CME information, but we've recorded your attendance and you will be getting CME for this talk. Please join us again in 2 weeks for Dr. Morgans is going to talk about hormone-sensitive prostate cancer. And again, Dr. Abdulkadir, thank you very much. Great work. I think it's very exciting, and I'm hoping to start seeing it maybe move forward clinically. I think it's on the horizon, so that's going to be wonderful.
Thank you.
Thanks, everybody.
Opportunities, like specific parts of the case. Halpern doesn't necessarily let residents do anyway. Like, you won't let a resident really do the first dilation for an IPP placement. And so I think he'll let Fantas do that, but I don't think it'll change and take away from what Halpern would let other residents do, although it'd be nice if residents were allowed to do some of those parts of the cases. But I don't think it would change how Halpern's currently teaching the residents, if that makes sense.
Okay. Well, I think what would be helpful to me is for us to put the expectations for the fellow, like of what we expect, what how you can contribute to the VA and to the residents' experience and education. Maybe the parameters. I think maybe if Emmy, if you'd send me kind of a brief summary, like email of the point that I will give to Brannigan and so that the fellow, so Fantas and Halpern is on the same, not in confusion about, what's going to happen, because that's what I don't want to happen. I don't want this to be a slippery slope where it turns into Halpern and Fantas doing cases together and the residents just, cutting suture. Like that's not going to be allowed to happen. But I think if we have it clear in writing, I think that would be really helpful. So could I ask you, could you put together a brief sort of email summary of what, you kind of the ideal things he could do to help would be for me, so I can share that? I think that'd be helpful to me.
I can do that.
Okay, and then I think Adarsh, it will be on, if Adarsh is on, I don't know if he is, really helpful that when this does start, that we monitor this, maybe, touch base once a week and, shoot me a one-liner email or whatever saying what happened on Monday, like how it went, because I told Brandon I'll be monitoring this really closely. And if there's any issues, then he can, I don't care what he does, but I don't want him to impact your guys' experience there. I really don't. And so that's my big concern. And then there's a new fellow starting in July. So I think if we have clear expectations and that fellow is also clear on what will happen at the VA, then everybody, it should be a win-win for everybody. But like I said, if it's negative for the residents in any way, I will fix it. So I just want to make sure that does.
That sounds good. I'll make sure it's followed through with.
Great. That's all I have. Anything else? Any other issues, concerns, things we can do for you? Okay, so is Neil on the line? I hope he's on. He should be, right?
I'm here.
Okay, so why don't you figure out if the residents want to do some sort of an outing? Like I said, kayaking, a picnic, I don't know. Trivia night, I don't know if anybody really wants to do any more Zoom crap at this point. And see if we can move forward with something if the residents want to do it. And then who wants to give me feedback about the food in the resident workroom, which is in Feinberg, not in Galter?
I can give you both.
Okay. Sounds good. All right, guys. Well, have a nice evening, and I look forward to hearing your ideas.
Sounds good. Thanks, Dr.
Thank you.
Have a good night.